Hello, I'm an applied ethologist who specializes in animal welfare. That means my work is to develop valid ways of assessing animal welfare and of improving it in practice. I work mainly with farm animals, but in a previous job, I worked on lab animals. I was focused especially on mice, which are the most widely used mammal in biomedical research. And all of our work on mouse welfare was also simultaneously about what I consider to be the major issue in lab animal science, and that's the crisis of translation. Translation here means transferring results from preclinical research on lab animals to clinical research on human patients. The problem is that the vast majority of results just don't translate. Overall, about 8 out of every 9 novel drugs that are tested in humans don't work, which is mind-blowing because they were all tested in animal models first, and they all seemed to work. It's also horrifying ethically because it means a huge waste of human and non-human subjects and patients who continue to suffer without effective treatments. So, why is this happening? Well, it's clear that this is a complex problem with multiple causes. A lot has been written on the subject. In this talk, I want to focus on making the case that the Strange framework can help explain the translation crisis. Translation is a form of generalization. We're trying to generalize results from a sample of one species, often the mouse, to a wider population of species that includes humans. The weird and strange frameworks are fundamentally about generalization. That first paper about weird people argued that what we know about human psychology is not universal, because our study populations tend to represent just one narrow slice of humanity. And more recently, other papers have made this same argument with regard to human medicine and related fields. For example, Girvin and Lieberman found that people from weird countries like Canada are about 40 times more likely to end up as a study participant in one of the top international medical journals. And there are important differences that impact the biology of weird people, including their susceptibility to disease and response to treatment. They tend to have lower pathogen load, less babies, be less physically active, and to consume more calories, more processed foods. With that in mind, let's go back to preclinical research. I'm about to argue that the mice we study also tend to be weird, or strange. They're not representative of mice as a whole. And if we can't even expect results to generalize from strange mice to other mice, or from weird humans to other humans, then we're in trouble when it comes to translating between species. This is an issue that I've been thinking about for a while. I've had this post-it note sitting above my desk for at least 4 years. I was trying to list out issues that make research mice non-representative of the species, and to then shoehorn them into an acronym like WEIRD or one of its synonyms. I wanted to do that because I'd seen the power of weird in psychology. It's become an effective shorthand for the whole idea that generalizability is poor because studies focus on a narrow range of subjects. Many authors have already written about this exact problem in lab mouse research. But it seems like the kind of issue that people constantly have to be reminded of, that they often ignore and hope that it will go away. So I very much liked the idea of having a powerful shorthand for mice, just like for weird humans. And after some brainstorming, it was my former grad school advisor, Georgia Mason, who eventually did come up with an acronym. And I think that will be presented in the following talk by Jessie Kate. So when the Strange Animals paper came out last year, I was a bit mad that I hadn't written anything yet, but especially I was just ecstatic to finally see the idea in print. And while the Strange framework wasn't built specifically for mice or for lab animals, I think it applies very well. So I just want to quickly show how some aspects of Strange apply in this case. First, the social background of laboratory mice is completely abnormal. In the wild, mouse social organization is quite flexible, but generally they live in some form of mixed-sex group. In the lab, they're typically housed in same-sex groups at high density. Especially in males, that can lead to severe aggression and chronic stress. The main alternative is single housing, which prevents aggression, but which is also abnormal for mice, which are a social species. Rearing history is also abnormal for the species. They're separated from their mothers at 3 weeks, earlier than weaning and much earlier than dispersal in the wild. Their physical environment is also very restrictive. It's a cage the size of a shoebox. Compare that to outside the lab, where mice have to navigate complex environments and usually maintain a home range of at least a few meters squared, sometimes much larger. Taking experimental measurements can often require that mice be handled, but the problem is that mice react to experimenters as predators, and that the stress caused by this inescapable aversive situation can interfere with outcome measures. For example, in pain research, stress-induced analgesia makes it so that outcomes can reflect how stress-sensitive a given mouse is, more than how pain-sensitive that animal is under normal circumstances. Most researchers still use the conventional handling method, which is to pick up mice by the tail. I've read several papers where teams try to habituate mice to tail handling, but nobody manages to do it with any consistency. Mice mostly continue to find this aversive, always. Finally, let's look at genetic makeup. Most researchers use inbred strains, within which individuals are more or less genetically identical. And there's one strain in particular, C57 Black 6, which is absolutely dominant. In some fields, like aging research, C57 Black 6 represents more than 80% of the animals used. It's difficult to find survey statistics on overall use of different strains, but I wouldn't be surprised if Black 6 are actually in the majority worldwide. In other words, a huge percentage of research mice, possibly even most mice, are members of the same very, very inbred family. And we know that it's not a particularly normal or average family either. Black 6 has long been the main strain used in pain research, but it turns out that there are outliers on almost every outcome measure typically used in pain studies. One study found that out of 11 inbred strains, Black 6 is the most unusual one. And it's also different in other ways that are nicely summed up in the article below my face. And some of these characteristics predisposed them to being chosen as models in some disease areas. For example, they're quite addiction prone. But those same characteristics also make them unusual among lab mice. And there are other issues that make the typical lab mouse a poor representative of mice as a whole. In a paper that came out in 2017, we summed it up this way. Imagine if clinical studies used patients that were all twin brothers of the same age with identical living conditions and whose house cleaner was a grizzly bear. All of these factors can affect outcomes in biomedical research, which often uses behavioral measures or measures which can be affected by acute or chronic stress. So, the research mice are extremely strange, and that makes it hard to generalize to mice as a whole. And the humans tested also tend to be weird. That means we often can't form an accurate picture of how mice respond to experimental treatment, and no accurate picture of how humans respond either. And that makes it doubly unlikely that results will translate from strange to weird. We're not weird in the same ways that the mice are strange. So, how do we fix this? I think one part of the solution is to treat mice more like human patients, and we argue for that in the paper on the bottom right. That would mean making their welfare a priority, giving them rich, complex lives, and having them participate in tests voluntarily, without coercion. And that would make mice a bit less strange, but the main translation benefit here would be that it would make them a different kind of strange, that gets them a bit closer to humans, for example by reducing effects of stress on experimental outcomes. The other option is to try to eliminate the strangeness, by going against what's been called the standardization fallacy. Instead of standardizing experimental conditions to isolate the one single factor we're interested in, like a novel drug effect, we could deliberately introduce heterogeneity into experimental designs. That means, for example, systematically varying strains, housing conditions, and other factors within an experiment. And that's already known to generate results that are more reproducible because they're less affected by the idiosyncrasies of each lab or each experimenter. And I'd argue that this would also improve translation, because sampling varied mice in varied contexts should give a more representative picture of mice as a species, or at least of lab mice as a group. And that's hopefully part of the solution to the translation crisis, along with using more rigorous methods and choosing appropriate models for each medical condition. Thanks to my postdoctoral advisor, Joe Garner, who turned me on to a lot of these issues, and to my graduate advisor, Georgia Mason, who helped me think through them more recently. Thanks also to lab members Gwendoline Caroui and Nicoleta Patamia, with whom I've discussed this recently. If you're curious to learn more, you can find full references and image credits in the YouTube video description below.